We'll go ahead and introduce our next speaker, Professor Daniela Weiskopf. You want to go ahead and start sharing your screen while I introduce you. Daniela received her PhD in immunology from Innsbruck Medical University in Austria, where her research focus was on post-translational modifications of virus-derived epitopes and modulation of the T-cell immune response during aging. She did her postdoctoral training at La Jolla Institute for Immunology, where she characterized human dengue virus-specific CD8 and CD4 T cell responses in samples from areas with endemic dengue infection and following experimental dengue vaccination. One important outcome of these studies was the development of dengue-specific epitope megapools that allow the testing of virus-specific T cells in small amounts of blood, irrespective of the HLA restriction of that donor and infecting serotype. So as a research assistant professor in the Center for Infectious Diseases and Vaccine Research at La Jolla, she's recently focused on the characterization of SARS-CoV-2 specific T cell responses. And she'll be talking to us about the immune responses induced by SARS-CoV-2 vaccination.
Thank you so much and good morning. So I'm excited to show you our most recent study where we have actually follow the immune responses that are induced by the different SARS-CoV-2 vaccinations. So, and I think I want to point out when I talk about vaccine protection against SARS-CoV-2, there is always different arms of the immune system involved. So if you typically think about detectable infection, protection against detectable infection, what people call sterile immunity, antibody is going to have a major role, and then T cells, CD4 and CD8 are going to have a minor role. But we know very well that SARS-CoV-2 infection and vaccinations also protect against more severe disease, hospitalization and deaths. And they're like all these arms of the immune system can have a part of it and do have a part of it. So that's why I think it's important to look at all of these different parameters of the immune system, not only antibodies, but also cellular immunity, because they have different parts in the clearance and of the virus and also protection against disease. So what we have been doing is we're looking at the COVID-19 vaccine platforms that have been deployed here in the United States. And of course, it's mostly mRNA vaccine. So the first one that we looked at was the mRNA vaccine developed by Moderna, which is shown here in red. The mRNA vaccine developed by Pfizer or BioNTech, which is shown here in blue. And then also what has been given here in the United States was that adenoviral vector developed by J&J. And this is shown here in green. And these are the colors that will also go through the remainder of my talk. Of course, you know that they have been given in different proportions. So the majority really has been given Pfizer and Moderna, and there's some people that received the J&J vaccine. So this has been the three constructs that we have been following throughout the study. And there's two different doses. So the mRNA vaccines have been given in 2 doses. The J&J vaccine has been given in 1 dose. So what are we interested in? What do we want to know is what is the immune response that is induced by these COVID-19 vaccine platforms? Because we thought it was really interesting because we know they all are given the same antigen. So all of them contain the spike protein of the SARS-CoV-2 virus, but they all are given in different vectors and different constructs. So is there a difference in the immune response induced by these different vaccine platforms? And we wanted to make sure that we do compare a direct side-by-side and comprehensively, because if you compare, if you study the vaccine responses against the first vaccine in 2022, and then the next one in 2023, and then you just cannot compare it side by side if you have not got it from the population, the blood from the same time, if you're not measuring it in the same machine, if you're not measuring it with the same person. So we carefully wanted to make sure that we take out as much of the variables before that we can really do a comprehensively side-by-side comparison. So this is basically our study layout. So you've seen here the vaccines that I have just quickly introduced. We did have later on in the study also access to some people that got the protein vaccine construct, which is shown here in purple, the Novavax protein. And this is only like we got volunteers that have been vaccinated and we got samples at the later time point. So we basically all collected the blood here at the clinical core, the La Jolla Institute. So all of these participants are collected in Southern California. They all have been processed by the same lab. And then we collected different baseline samples. Then that time point 2 was like 2 weeks post vaccination, 42 days post vaccination. Then we were interested in like, what about 3.5 months out of the time point? This time point has been chosen because like traditionally, if you look at these vaccine studies, a lot of people look at this and then report on like, the shrinking of like the repertoire. So we wanted to see like, what is it like when you look at 3.5 times of 3.5 months after vaccination. And then also time point 5 is like 6 months after vaccination. And we think this is an important time point because you can actually look at the true memory that has been induced by this vaccine, because you have all the effector functions being contracted by that time. And as I pointed out, we look at all arms of the immune system, the cellular immunity, B cells, CD4 and CD8 T cells, but also antibody responses. For the reasons that I presented in the first slide, I think all of these parts of the immune system have different parts to do. So this is the characteristics of our COVID-19 vaccinees. So you can see that the donors, we collected 30 from each of these vaccine cohorts, and I told you like Novavax came in later, so you can see also like later time points have been collected from that. Overall, the male-female ratio is comparable between these groups. The age is comparable with these groups. They are all like approximately 40 to 45 years old. And also like the race or ethnicity is comparable here. And I think that's important because we made sure that we not, look at one vaccine group that has older participants, one that has younger, so that the difference in vaccine response is not due to like, any of these factors. So comprehensive recruitment of these vaccinees. And this is just to show you a different way of like, when is the time points that we have been collected. So you can see here the different time points that we've just been going through T1 to T5. And you see that for all of these vaccines, we have some some donors that we collected all time points, some that we collected later time points. But overall, we get like a representation of like pre-bleeds and also like after 6 months. And that's true for like all the vaccines, with the exception of Novavax, which I pointed out, we don't have the earlier time points. We have the 3.5 months and the 6 months time points. And what did we find? So I will be going through like the different parameters of the immune response. And as I said, like the color code is always the same. So red is the Moderna vaccine, blue is the Pfizer BioNTech vaccine, green is the J&J vaccine, and the purple responses are the Novavax vaccines. And so if you're interested in details, like all of this is like shown here on the top of the graph. So you can see how many samples we measured for each time point, how many people did respond. That means like we consider this a positive response. And then also like if there's any difference between any of the other vaccine groups, statistical difference. So if you want to see the details, you can always check here on the top of the graph. So the first thing we were looking at were like RBD-specific IgG antibodies. And that is because like that is like, like the one thing that the majority of like vaccine developers and also like people measure. So, and we saw previously that there's a different kinetics in these mRNA vaccines compared to adenovirus vector vaccines. You can see there's a quick peak after the first dose, it goes higher after the second dose, and then it declines towards the memory phase. And that is very comparable between like both of the mRNA vaccine constructs, different to the adenovirus vector construct, where we can see, remember, this is only one dose, you see an increase, and then it continuously keeps increasing, not to the same magnitude as with the mRNA vaccines, but you can see after 6 months, you can still see a response in 100% of the people that have received this J&J vaccine. What about the protein vaccine? You do see a strong induction of antibody responses at the later time points, and that basically remains stable between 3.5 and 6 months. we were interested then also like what is the functional response that is induced, because you can measure IgG titers, which gives you a level of the magnitude of antibodies that are induced. But if you want to know if your antibodies are functional, you measure typically neutralizing titers. So we have been doing that too, to compare the different vaccine constructs and also to get a more functional readout. And this is basically shown here. So again, just similar following like the IgG ELISA titers, you see a strong induction of neutralizing titers after 2 doses of the mRNA vaccines, and that declines after 6 months, but still very much detectable, like in 100% of the donors in 2 of these mRNA vaccines. Similar to like the IgG titers, you also see an increase in functional neutralizing titers with the adenovirus vector vaccine. And you can see again, the level is lower than compared to mRNA vaccines, but you do see an increase all the way up to 6 months. Strong induction, but a lot much broader like variety in the protein vaccine, but you see there is neutralizing titers in 90% of the people 3.5 and 6 months after vaccination. So how does this compare to like a natural infection? So we have measured people at the same time point collected that have been infected 6 months out of after like natural infection. So these are all shown here in the yellow spots. So these people have been infected, and we collected blood at a comparable time point when we actually looked at all these antibody titers. And you can see that there is no difference between antibody titers induced by the mRNA, between the adenovirus vector and the protein vaccines, and there's slightly a stronger induction by mRNA vaccines compared to natural infection. This is true for Moderna and also the Pfizer BNT vaccine. Similar picture for like the neutralizing titers. Again, there is like slightly stronger induction with like the mRNA vaccines of neutralizing titers, and comparable with like the other 2 vaccine constructs. So in conclusion of the serology part of this study, we showed that COVID-19 vaccine platforms exhibit distinct peaks of antibody responses, and they are different between like mRNA vaccines and the viral vector vaccines. We do see a strong response induced by like the mRNA vaccines that peak 2 weeks after second dose, but still very much detectable after 6 months of vaccination. So overall, we see a strong induction of these IgG, RBD, and also neutralizing antibodies that are durable for up to 6 months. And that is different levels for the different vaccine constructs, but overall, we do see that for the majority of vaccines. What about cellular immune response? So we have been looking into the B cell response, and this is measured spike-specific B cell responses, as you can see here, measured by 2 different the spike protein tagged with 2 different fluorochromes. And you can see here that there is a strong induction of the memory B cells, and which is detectable with like the construct of these 2 different protein fluorochrome measurements. And if you look at the spike-specific B cell responses that are induced by these different vaccines, you can see that regardless of the vaccines, we do see a strong induction of memory B cell response, which increases over 6 months of post-vaccination. And this is why I show you the later time points here. And this is also not shown any difference between like memory B cells that are induced by natural infection. So overall, regardless of the vaccine constructs, you see very similar induction of spike-specific memory B cells, regardless of the COVID-19 vaccines. That does increase over the time points of vaccination. So we were then next interested in the T cell response, and this is basically just summarizing what I just said. We do see frequently detection of memory B cells, and we saw the IgG isotype is like the most dominant population, which I haven't shown you the data, but it is in the publication. So T cell response is the next arm of the cellular immune response, and here we measured the spike-specific T cell response with like the megapools that we have been designing. So basically the megapools in this case is like overlapping spanning the entire spike protein. And we do this because in this case, we don't have to know the HLA phenotype of these people. We can use the overlapping spike-specific T cell response. And by having 15 mers overlapping by 10, spanning the entire spike protein, we are able to detect regardless of the HLA phenotype of this donor. And it's also something that is useful when you just want to get a quick general overview of the antigen specific response in your population. And you can see here that again, if you look at the 4 different vaccines, there's a strong induction of CD4 T cell responses in the mRNA vaccines. It is strong increase after the first dose, goes even higher after the second dose, and remains somewhat stable with a small decline, 1.5 time decline after the 6 months memory time point. So strong induction of memory CD4 T cell responses. And that is the case for both of the mRNA vaccines, which only slightly decline in the memory phase, as I pointed out, that at a 1.5 time point lower than after peak of the response. So interestingly, the J&J vaccines follows a different kinetics. So you do have a strong induction after 1 dose only, not to the same level as the mRNA vaccines, but you see a strong maintenance of the response up to 6 months after 1 shot dose. We do see a strong reduction also with the protein vaccines on CD4 T cell responses that does not drop between 3.5 and 6 months time point. So if you now ask how does this compare with natural infection, you see a very comparable induction with the natural infection between the different vaccines constructs. The only one that is slightly significantly higher than natural infection is in immune response, CD4 T cell response induced by the Moderna vaccine. Again, just that we looked at for the antibody responses, we're not only interested in the magnitude of responses induced, but also in the multifunctionality. And in this case, we are looking into the ability of these antigen-specific cells to induce cytokines. And this is basically showing you the number of functions that are induced, and we measured a broad spectrum of Th1, Th2, IL-10, IL-17 cytokines, so that we can get an idea of not only the magnitude, but also the functionality of the T cell response. And you can see here, similar to what we see for the AIM assay, we can see a strong induction of multifunctionality and T cell response that is then also maintained with a small drop, 2-fold drop 6 months out after vaccination. And we did see a slightly slower response, a lower response of the Pfizer vaccine, but you can see again the same kinetics if you see a strong response followed after 2 response. Interestingly, you see there's a lot of people that needed the second shot to induce a stronger immune response, CD4 T cell response functional after 2 shots of vaccination. We do see an induction of spike-specific CD4 T cells with the adenovirus vectors, but it's a much lower level, as you can see here. And we do see also maybe only 60 or 43% of the people do still have a response of CD4 T cell response 6 months out after vaccination. So that is different to the parameters that we've been seeing before. So we do see differences between the vaccine constructs in regards of multifunctional CD4 T cell responses. We do see an induction of multifunctional CD4 T cell response also with the Novavax response. Again, no difference between the memory time points, 3.5 and 5 time points. And if you compare it to what is induced by natural infection, again, much stronger multifunctional immune responses induced by the Moderna vaccine constructs, comparable to all the other vaccine constructs if you compare it to natural infection. So if you're interested in tying this together, so if there is an induction of follicular helper T cells, this is what we typically measure, because that can give us an idea of is there a CD4 T cell responses induced, and I can help antibody responses. And we do measure T follicular helper cells for all of these vaccine constructs, and you can see again follows a similar kinetics between the mRNA vaccines and is also like a lot more like different between the J&J, about 2/3, a little more than 2/3 induced T follicular helper cells received by that adenovirus vector vaccine. We do see also 100% of the people do have T follicular helper cells after the protein vaccines. So again, this is something that we are interested to compare with natural infection. And interestingly, we do not see any difference in the induction of T follicular helper cells, regardless of the vaccine constructs. Finally, the last part of the CD8 T cell responses is CD8 T cell responses. And so you can see here, we do see an immune response induced by all of these different vaccines, antigen-specific CD8 T cell response. And all of this is again showing you the multifunctional readout. And you can see there is a similar kinetics than with the CD4 T cell response, much lower level of magnitude, but they do maintain in the memory phase with a smaller drop, about 2-fold drop after 6 months of infection. And again, for the CD8 T cell responses, we do see like a stable induction, a lower level than compared to the mRNA vaccines, but this does not drop as much as we do see for the mRNA vaccines. Interestingly, we barely saw an immune response in 10% after the first dose, and in about 0.5% after like 6 months, we do see follicular helper cell response by the protein vaccine. So there's a difference in that. So overall, I can summarize this. We do see spike-specific CD4 and CD8 T cell responses induced by the different vaccine platforms, with a difference of like the Novavax protein that this induces like a much lower level than the mRNA vaccines. We do see a different distinct peaks of the spike-specific T cell responses depending on the vaccine given, and we also see follicular helper cells induced for the COVID vaccines that we've been looking at. If you have a data set where you measured all these parameters over these time points, you can also ask the questions like, what does correlate with each other? So we asked the question, can we like look at 3.5 months after vaccination and see what correlates with like the 6 months time point? And we can see a strong correlation of like the CD4 T cell responses that correlates with the antibody responses 6 months after like vaccination for the mRNA vaccines. But that was very interesting, like there's zero correlation of these parameters with the adenovirus vector vaccine. So it seems to be a much more like orchestrated immune response induced by the mRNA vaccines compared to the adenovirus vector vaccine. So, but this is just to get to show you like this is like the power of a data set like this, where you have like all these different parameters measured at all these different time points, so that you can ask what is like correlating with what. And so this is interesting because I think there's a strong difference depending on the vaccine construct, with depending on what the vaccine that has been used. So overall, I've been showing you that there's early spike-specific CD4 T cell response that correlates 6 months after vaccination. In the mRNA vaccines, does not so with like the J&J vaccine. And it's not, we cannot really do these correlations with the Novavax vaccine because we have a much smaller cohort size. So that's why I've shown you only these two different correlations. So overall, this is basically summarizing the entire thing that I've been showing you. This is a head-to-head vaccine comparison spanning from like pre-bleeds to 6 months after vaccination. We compared it with like natural infection, and I pointed out that this is like mild COVID-19 vaccination. So we don't know if there would be a difference if we would have people that had like a more severe form. And this is just basically like all the points that we have been going through one by one, and just showing you like the difference and the similarities induced by the different vaccines. And with that, I'm just going to thank all the different groups that have been involved. So on the right side is the people from my group, but of course, strongly involved also Alex Setter's group and Shane Crotty's group that have been involved in this study, because it takes a lot of people to measure all of these different parameters. And with that, thank you for the invitation, and I'm happy to take any questions if you have any.
Excellent. Thank you so much, Daniela, for that very impressive data set. We do have time for maybe 1 or 2 questions.
Yeah.
Jeremy, yeah, go ahead.
Very quick question. Thank you very much, Daniel. That was a great talk. with regards to the CD8 T cell detection that you were doing there, were you still using the 15 megapools, or were you using a slightly different strategy?
Yes, no, this is also like using the 15 megapools, and we have carefully titrated the time points to make sure we can still see CD8 T cell responses with this method. I do have to point out, so what we don't know is like, because we see CD8 T cell responses at some time points, they go away at some time points, come back at some time points. So, I mean, there's a strong suggestion that they might be going like somewhere in the tissues, and we don't always see this in the periphery. So, so we think we have titrated this, like the time course for this same method, so we do know we can identify CD8 T cell response, and it's not bystander response because we have been depleting CD4 T cell responses to make sure. But yeah, so of course we are relying on peripheral blood, so it could be different if we would like go like somewhere in the tissues and do like lavage or something.
Yeah, thank you very much. Thank you.
Right, Professor Shin.
Yeah, thank you. So once in the John Wary group, they define the immune durability after COVID-19 vaccination at a relative level of 6 months titer compared to peak titer. So having to compare this the mRNA vaccine and J&J and protein vaccine in terms of durability, 6 months durability in terms of antibody or T cell response, all the immune parameter. Which one was much more durable?
Yeah, so all of these time points, T5 time points was always 6 months. And so you see stronger induction of like because you start so much higher, right? The kinetics for the mRNA vaccines are much stronger. So the memory response is also higher at the 6 time points. What is completely different is the kinetics, because like you give them one shot of the adenovirus vaccine and you do see like a maintenance of the memory, but the level is much lower than when you give them the mRNA vaccines. Yes.
Okay.
Excellent. All right. Well, thank you again to all of our speakers in this first session. Those were fantastic presentations.